Dear friends, my name is Mr. Desley SD. I am assistant teacher in Mahatma Gandhi Vidya Mandir's Janta Vidyalay and Junior College Manur, Taluka Kalwan, District Nasik. My subject is biology. We are going to study now chapter number 4, molecular basis of inheritance. It is our third lecture. Start from the substitute the different topics and completed these chapters, that is molecular basis of inheritance. In this lecture, the first topic is regulation of gene expression. It is the multi-step process by which a gene is regulated and its products is synthesized. Thus, the gene expression result in the formation of polypeptide genes. Expression process is known as regulation at different levels. In eukaryotes, the regulation can be at the different levels. The first level is called as transcription level. Second level is called as processing level. Third level is called as transport of messenger RNA from nucleus to the cytoplasm, and fourth level is translational level. All these four different levels completed the regulation of gene expressions. In this regulation of gene expression, the two scientists, that is Jacob and Monod, in 1961, on the basis of their study on the inducible system of the synthesis of beta galactosidase enzyme in E. coli, proposed a model to explain the induction or repression of enzyme synthesis. These two scientists, Jacob and Monod, shows that regulation gene, promoter gene, operator gene, and structural gene. These four types of genes are present in regulation of gene expression. In these four types of genes, the main type of gene, that is called as structural gene, and this structural gene consists of three different types, that is Z gene, Y gene, and A gene. The second topic in this lecture, that is an important topic called as operon concept. It is the transcriptional control mechanism of gene regulation, that is functional unit of DNA containing cluster of gene or genes under the single promoter with the help of operator. Francisco Jacob and Jacques Monod explain that metabolic pathways are regulated as a unit. For example, in E. coli, when lactose sugar is provided to the culture medium, cell-induced production of three different types of enzymes are necessary for digestion of lactose, and these three enzymes are: number one, B galactosidase; This type of enzymes digest lactose into galactose and glucose. Means beta galactose are important enzyme in the digestion of lactose. The second type of enzyme, that is beta galactoside permease, they permits lactose molecule to enter into the cell. Means lactose molecule enter into the cell with the help of beta galactoside permease enzyme. Means the entry of lactose depends upon beta galactosides permease into the cell. And third type of enzymes, that is transacetylase. This type of enzyme transfer acetyl group from acetyl coenzymes to the galactosides. Means all these three types of enzymes are important in this process of operon concept. The synthesis of these three enzymes is controlled by a long segment of DNA known as operon concept. It consists of an operator site O and three structural genes, that is Z gene, Y gene and A gene. The action of the structural genes is regulated by the operator site with the help of repressor protein. Repressor protein is produced by the action of genes I known as regulator genes. Next topic is lac operon. They consist of following compounds. Number one, regulating genes or regulator genes. It is also known as repressor genes. Number second, promoter genes. Number three, operator genes. Number four, structural genes. And number five, inducers. These five different types of components shows the lac operon concept. In this lac operon concept, the E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into the sugar used for cellular metabolism. Means these five different types of genes are used by lactose may be converted or lactose may be break down into the sugar. Means these components are important for breakdown of lactose and it is converted into sugar. These five types of genes, that is number one, regulator gene. These genes control the operator genes in the cooperation with an inducer present in the cytoplasm. Regulator gene proceed the promoter gene. It may be not present in immediately adjacent to the operator genes. Regulator gene producer a protein called as repressor protein. Repressor bind with the operator gene and repress its action. It is called as regulator proteins. The second component, that is promoter gene. This gene proceed the operator gene. It is present adjacent to operator gene. The promoter gene marks the site at which the RNA polymerase enzyme binds. When the operator gene is turned on, the enzyme moves over the operator gene. Promoter gene base sequence determines which the strand of DNA acts as templates. Number 3, that is operator gene. It precedes the structural genes. This control the functioning of structural gene. It lies adjacent to the structural gene. When operator gene is turned on at the inducers, the structural gene produce messenger RNA. Operator gene is turned off by product of repressor genes. And last, number 4, that is structural gene. It may be consist of Z, Y and A genes. Lactose is added to the E. coli culture. The structural gene catalyzes messenger RNA, which in turn produces polypeptide on the ribosomes. These polypeptide forms act as enzyme to catalyze lactose in the cell. There are 3 structural genes in the sequence of LAC-Z, LAC-Y and LAC-A, means lactose Z, lactose Y and lactose A. Next topic, that is role of lactose. The role of lactose, the few molecules enters into the cell by an enzyme permease, and this lactose may be converted into sugar, and the sugar are usable content into the cell. A small amount of enzyme is present even when operon is switched off. A few molecules of lactose act as inducers and bind to repressors. This repressor inducer complex fail to join with the operator gene, which is then turned on. Structural gene produces all enzymes. Thus, lactose acts as an inducer of its own breakdown. When the inducer level falls, the operator is blocked again by repressor. So, structural genes are repressed, inactivated again. This is negatively feedback. It is the role of lactose. And next, an important topic, that is genomics. In 1920, the scientist H. Winkler is the total genetic constitution of organism called as genome. Means the total genetic constitution of gene introduced the scientist H. Winkler, and this term is known as genomics. All organisms contain this important constitution of genetic material. Alternatively, it is a complete copy of genetic information, that is DNA, or one complete set of chromosome, that is monoploid or haploid of an organism. The term genomic, coined as T.H. Roderick in 1986 also, it is the study of genomics or genomes through analysis, sequencing and mapping of genes, along with the study of their functions. The various applications of genomics are present in the science, that is, structural and functional genomics is used for different purposes in the improvement of crop plants, human health and livestock. The knowledge and understanding acquired from genomics research can be applied in a number of different sectors, including medicine, biotechnology and social science. It helps in the treatment of genetics disorder through the gene therapy. The following application, that is, number 1, genomics is used in agriculture to develop transgenic crops having more desirable characters. Number second, genetic marker developed in genomics have application in forensic analysis. And last, number three, and main application, that is genomics can leads to introduce few crops in microbes to produce enzyme, therapeutic proteins, and even biofuels. Next topic, an important topic, that is human genome project. In human genome project was initiated in 1990 under the International Administration of the Human Genome Organization, the short form, that is HUGO. The project was coordinated by the US Department of Energy and National Institute of Health. Human Genome Project are important by following aims, that is mapping of entire human genome at the level of nucleotide sequences, to store the information collected from the project in databases, to develop tools and techniques for analysis of the data, transfer of the related technologies to the private sector such as industries, and last, taking care of the legal, ethical, and social issues which may arise from projects. And hence these aims, this important aim shows the mapping of entire genome and at the level of nucleotide sequence present in human genome organization. And these projects are important in health of all living organisms. Next topic, that is DNA fingerprinting. Genes are important into the cell, but this type of gene present on chromosomes, and these chromosomes are responsible for the determining character of organisms as well as inheritance of characters, and hence gene is known as structural and functional unit of cell. Means all types of genetic characters are inherited with the help of genes, because DNA means deoxyribose nucleic acid present in genes. Due to the recombination of parental and maternal genes, we differ from our parents. Difference also arises due to the infrequent mutation that occurs during the gamete formation in the process of cell division. Due to all these factors, every individual has its unique genetic makeup, which may be called as fingerprint. The technique developed to identify a person with the help of DNA restriction analysis is known as DNA profiling or DNA fingerprinting. The technique of DNA fingerprinting was first given by the British geneticist, Dr. Alex Jeffrey, in 1984. And these all the functioning, all the character of DNA fingerprinting, all the process of DNA fingerprinting as shown into the figure. And by the process of this DNA fingerprinting, they develop to identify a person with the help of DNA restriction analysis. The following steps involves or aims involves in DNA fingerprinting, they are as follow. These are 7 different aims involves in the DNA fingerprinting. First one is isolation of DNA. Second one is restriction digestion. Number 3, gel electrophoresis. Number 4, southern blotting. Number 5, selection of DNA probes. Number 6, hybridization. And number 7, photography. All these 7 steps, the DNA fingerprinting complete process, they identify the parents of the particular organisms. That is number 1, isolation of DNA. The DNA must be recovered from the cells or tissue of the body or host. Only small amount of tissue like blood, hair roots, skins, etc. is required. Number 2, restriction digestion. The isolated DNA is treated with restriction enzyme. This enzyme cuts DNA into the small fragments having variable length. This phenomenon is called as restriction fragments length polymorphism. Number 3, gel electrophoresis. The DNA samples are loaded for the agarose gel electrophoresis under the electric influence. The DNA fragments which are negatively charged move to the positive poles. The movement of these fragments depends on length of the fragments. This results the formation of bands. DNA splits into the ssDNA by alkali treatments. Number 4, southern blotting. The separate DNA fragments are transferred to a nylon members or a nitrocellulose filter paper by placing it over the gel and soaking them with filter paper overnight. Number 5, selection of DNA probes. A known sequence of single-stranded DNA is prepared. It is called as DNA probe. DNA probe is obtained from organism or prepared by cDNA preparation method. The DNA probe is labeled with radioactive isotopes. Number 6, that is hybridization. This probe DNA is added to the nitrocellulose filter paper containing host DNA. The single-stranded DNA probes pairs with the complementary base sequence of the host DNA strand. As a result, DNA-DNA hybrid are formed. Number 7, photography. The nitrocellulose filter paper is photographed on an X-ray film by autoradiography. The film is analyzed to determine the presence of hybrid DNA. And last topic, that is applications of DNA fingerprinting. DNA fingerprinting shows various types of applications into the science or biological environment. Number 1, in forensic science, DNA fingerprinting is used to solve the problem of rape and some complicated murder cases. Second application is, it is used to find out the biological father or mother or both of the child in case of disputed parentage. Last one, that is DNA fingerprinting is used in pedigree analysis in cats, dogs, horses and human. Thank you.